Hello and welcome everyone for this podcast for Epigenetics. In this podcast, we're going to talk about epigenetics and memory. Understanding the role of epigenetics with memory is probably the most difficult thing to study in the field of epigenetics. Understanding how your personality is, different kinds of behaviors, memory, how one learns, studying the aspects of disease and memory as it's linked to epigenetics. Turns out to be really difficult to study, but it's also maybe the most important thing to study. The author of your book makes this statement. I want to write it down because I really like it, and I think it sums up my feelings about memory and why it's important. And he writes, the retention of memories is the basis of our personal identities. Because the way we perceive the world, the way that we see things, we hear things, we taste things, we listen to things, shapes our memories. And those memories, in turn, determine, or at least help determine, who we are as a person. So I really like that statement. So we don't know as much about this as we would like, but I want to talk to you a little bit about what we know about memory and some of the links to memory and epigenetics. So let's first talk about short-term memory. Sometimes we might call this our primary memory or our active memory. In general, we can think of our short-term memory as the way we process current information that stimulates our senses. The part of our brain that is important in short-term memory is going to be found in this prefrontal cortex. More specifically in the prefrontal cortex, it's the supragranular pyramidal neurons in the dorsolateral prefrontal cortex. This part of the brain is highly developed in primates, more so than in other animals, which is one of the many things that makes primates distinct from other mammals. So if memory is going to be stored here, or at least short-term memory is going to be stored here, how are those memories made? What's the cellular context of memory? So let's talk about that next. So how is a memory made? How do I remember what I ate for breakfast? How do I remember my phone number? How do I remember the song that I heard when I was a kid and I still know the lyrics to today? How do I remember that childhood memory when I was 5 years old? How do I remember these things? I find this to be one of the most impressive things about our brain, that we have the ability to maintain these memories. It all starts with neuronal communications. So we have one neuron here and one over here. I guess I can draw the rest of it, or at least part of the rest of it here. But these are going to be, or at least can be, very long. They can be up to 1 meter in length, these neurons. They can also even have extensions, these dendrites coming off of them, the neurons. This would be considered the cell body. This is where you'd have your nucleus. We would have the cytoskeleton extending down all of these, so that vesicles made in one part of the nucleus or obtained from a preceding neuron can pass those messages along, or those vesicles along. These microtubules will help produce these extensions from here, the dendrites. So they're an important part of this neuronal complex. On this side we have the presynaptic neuron, and over here we have the postsynaptic neuron. Here, in between the pre and postsynaptic neuron, we have the synaptic cleft, the space in between these two neurons. Now what happens is when this neuron, or its preceding neuron, is stimulated, it will send a message along this neuron that allows the membranes of these plasma membranes to depolarize. So we'll have various ions like calcium or potassium or sodium that will move from the presynaptic neuron into the synaptic cleft and then finally into the postsynaptic neuron. So this flow of ions allows the membranes to depolarize, which generates and electrical potential difference. Every time the neuron is stimulated in the same way, maybe it's the smell of bacon that you smell, and it will stimulate the neuron in a certain way. And every time you smell bacon, it'll do the exact same pathway. But a memory isn't established just by depolarizing some of the membranes in one neuron. Rather, a memory is a collection of neurons depolarizing in a specific pattern. When that same stimulus is received by the neurons, it will stimulate the same collection of neurons. Over time, it establishes that short-term memory, and eventually it will lead to a more long-term memory. So different memories have different neuron circuits. When I smell my favorite food, that sense signals a different neuronal circuitry as opposed to when I hear my favorite song. Remembering a phone number, remembering all these things that we have crammed in our brain are established by having a different set of neuronal circuitries. So the electrophysiological activity that we see here that is initially established during short-term memory, over time will lead to that long-term memory. Now, we said the prefrontal cortex was really important in that short-term memory, but ultimately those long-term memories are stored somewhere else in our brain. So the hippocampus here is needed to establish long-term memories. We know that if the hippocampus is damaged, that these individuals can't establish new long-term memories. They'll still have old long-term memories, because what happens is that long-term memory is set in the hippocampus, but it is stored somewhere else in the brain. So they lose the ability to make new long-term memories, but other parts of the brain, like the dentate gyrus that I've put an asterisk by, are responsible for some of that storage of those longer-term memories. How long it takes to go from here to here, we don't know for sure, but we know it's not quick. We know it takes a considerable amount of time. So let's talk now about forming long-term memories. We're still learning quite a bit about forming these memories, but several studies have highlighted some structural changes at this neuronal junction here between the pre- and postsynaptic neurons. One such study took several rats. Some studies did mice, some did rats, and they had these rats develop specific tasks to get their food. So they might have had to go through some kinds of obstacles, they had to go through certain barriers. Different tasks had to be performed before they got their food. Now, if the memory is just getting your food and it didn't matter tasks, the neuronal circuitry and the architecture of it should be the same. But what they found was that with the different tasks, each rat established a different neuronal circuitry. And what they found was a difference in two different things. One was that the number and size of vesicles at the presynaptic neuron were different. So I didn't describe this before, but what happens is various vesicles will form in the body of this neuron, and they will travel along the microtubules and reach the synaptic cleft once the neuron is stimulated. And depending on the kinds of tasks these different rats did, they would have a different number of vesicles at the presynaptic terminal here, and different sizes of the vesicles, suggesting that different stimuli caused by these different tasks allowed different kinds of vesicles to form, and different amounts of those vesicles. Another thing they noticed that was different was that the cytoskeleton was different in the postsynaptic neuron. So what you may see extending, sometimes even from the axon itself, were protrusions coming out, but more often they were found here during on the dendrites, where they have these long protrusions sticking out, known as dendritic spikes. But these are dendritic spikes. Now these dendritic spikes now can interact with other neurons, allowing for more connections and more circuitries to be established. Those different circuitry that were established were unique to the different tasks that were performed. And the reason that we see these, to link it back to the cytoskeleton, is that these protrusions, these spikes, are established through the use of the cytoskeleton extending into the plasma membrane and pushing it out, with the end result of it establishing more connections with other neurons. So this helps explain how these different circuitries could be made and ultimately can lead to this long-term memory, based on the fact that they saw these in areas of the brain that are linked to long-term memory generation. So an idea was put forth that these neuronal changes reinforce connections and increase the ability of the postsynaptic neuron to respond to the presynaptic neuron. So in order to be able to respond appropriately to these different vesicles, the ability for these vesicles, or neurotransmitters that enter into this cleft here, to bind to this postsynaptic neuron requires that the postsynaptic neuron has receptors here, and always has receptors here, so that it can respond to this stimulus that's generating these circuitries. So this is one of the problems that is presented, that we always have to constantly be producing these receptors. So the genes that are going to upregulate these receptors and maybe turn off other receptors has to be established in the nucleus. And this opens the door here to some gene regulation, but also our good friend epigenetics. So now I would like to steer this discussion towards epigenetics as we move towards our discussion of epigenetics and memory. We've described how a memory can form, or at least what we think about the way it could form, and we ended on the last whiteboard with the idea that we had to have a way to change the nuclear transcription machinery so that certain receptors could always be made to respond to those environmental cues that are establishing that memory. So this gene expression, as I alluded to on the last slide, certainly has some standard mechanisms of gene expression. Getting the transcription factors there to turn on specific genes, bringing the RNA polymerases, and all that good stuff. But since these memories are being established by environmental cues, it's also quite possible that there's going to be an epigenetic component of memory. Let's hope so. If there's not at least a little bit of a hint of one, I just wasted a lot of time talking about memory. But there is, luckily. I want to begin by talking about a particular channel here on a postsynaptic neuron. In this example here, we're going to have calcium in this synaptic cleft. And it wants to get in here, but ions, if you remember back from an earlier biology class, have a very difficult time getting across membranes. To get across a membrane, ions need some sort of a channel in order to get across the membrane. And the particular channel I want to talk about here is a channel called NMDA, which is a glutamate-gated ion channel. So as long as there's no glutamate, calcium is not going to get through this channel. But once glutamate binds to this channel, what happens is the proteins will have a conformational change, opening up this channel, allowing calcium to pour in here. So there is some signal given to the presynaptic neuron that releases calcium, and then once calcium is in this presynaptic cleft, based on this signal, these glutamate molecules will bind to this NMDA channel, open it up, and calcium will have an influx into this postsynaptic neuron. Now, this calcium is a very important second messenger for signal transduction. So we know that calcium is used in many signal transduction pathways. In this particular case, what calcium will do is it will move to the neuron body here, and it will enter the nucleus. I'm going to draw a big nucleus here because there's a few things I want to draw on here. The nucleus really proportionally is not that big. But through various signal transduction pathways, a gene in here called ERK, E-R-K. And ERK stands for extracellular signal regulated kinase. So calcium, through these signal transduction pathways, will cause the ERK gene to be transcribed, and it will eventually then be turned into an ERK protein in the cytoplasm. ERK then will come back into the nucleus, and in the nucleus it will activate many genes. So these purple genes here are various transcription factors, and these transcription factors will in turn activate genes that we know are linked to memory. Now, as I said, some of this is just our standard mechanisms of gene expression. But it's not difficult to imagine that chromatin structure is also going to be important in order to maintain a more permanent gene activity within these cells, as these circuitries have been established in a certain way to signal certain pathways. So we don't know all of the epigenetics that are linked to it, but there have been several experiments that have shown changes in acetylation and methylation, and I want to just highlight some of those experiments with you. So let's begin with the experiments that show a link with acetylation. If you expose mice to a light pulse and then we look at their hippocampus, we see more acetylation on H4s, histone 4s. What genes they're linked to, we don't know yet, but we see more acetylation. Stimulating a fear factor, we see in the hippocampus more acetylation on H3, but not H4. The difference, we don't know what's happening there and why that's important in the gene expression, but these two pieces of evidence suggest that by stimulating the senses of the mouse under different circumstances, we get a different response to the chromatin. In yet another experiment, if you use HDAC inhibitors, and this first one is an in vitro experiment, so outside of the mouse, they measure an enhanced sensitivity of hippocampus neurons. They had increased electrochemical signaling of these neurons in the hippocampus. Now, if we also took HDAC inhibitors and did an in vivo study with them, so gave the mice these inhibitors, they saw, and this is pretty interesting I think, enhanced long-term memory. They were better at remembering things with these HDAC inhibitors. In other studies, they showed that acetylation in the hippocampus is relatively short, like 1 to 2 hours. It's reasoned that what's happening here is various HDAC proteins come along, the deacetylases, and compete and begin to remove these acyl groups, which goes in nicely with these data here saying that if you give them HDAC inhibitors, we see more acetylation and we see this enhanced long-term memory. Next, let's talk about methylation. So histone methylation. And in this study, they did a fear response, and let me describe what that is. I should have done that probably on the last slide when we first talked about it. But they would take a mouse, they would put it in a new environment, and then in this new environment, they would apply a fear stimulus. This is sometimes established by having puffs of air go in its face, or they may put short shocks to the foot to establish a fear response. They then remove the mouse, and then after a certain amount of time, they put it back into the same environment. If they have a memory of this fear stimulus, they should react in the same way as when they had that fear. And that's what happens here. When you put them back in the same environment, they freeze in one spot. Now what they showed here was that after a 1 hour fear shock, they saw an increase in H3 K4 triple methylation. And remember from back earlier in the semester, this is a sign of activation of a gene. Interestingly, they also saw an increase in H3K9 methylation. And if you remember, that is a sign of repression. Now, when they did these studies, they were studies and they just examined the whole chromosome, or all the DNA from the hippocampus. And all the DNA in the hippocampus, they saw more H3K4 methylation and H3K9 methylation. It doesn't mean the same regions had this. It just means overall. So these results suggest that in establishing this memory, some genes are upregulated and some genes are downregulated. They don't know which genes are which yet. That's the next step in these experiments. Now, something else interesting that they did with these studies is that after this 1 hour of the fear shock, methylation patterns decreased. So they shocked them, they waited about an hour, and then when they look at the methylation patterns, they're gone. Then, and this was a very interesting result, at about 48 hours, the dentate gyrus had increased H3K4 methylation, the activating kind of methylation. The dentate gyrus is activated or communicates with the hippocampus in establishing long-term memories. So the idea is that this initial fear shock establishes this short-term memory and begins to prepare it for a long-term memory that is eventually set in the dentate gyrus. All right, the last thing I want to talk about is Alzheimer's disease. And I'm not going to spend a lot of time on Alzheimer's disease, but I think it's appropriate to at least mention it here. We're talking about memory, relevant to talk about a disease that's linked to losing your memory. We know that Alzheimer's disease is the most common form of dementia. We know that it's caused by an accumulation of beta amyloid protein aggregates. And these aggregates will form on the neurons. However, only a small percent of cases, and I don't know the percent off the top of my head, but a small percent of the cases are directly linked to a specific gene mutation. Some are, particularly when you consider the early onset form of Alzheimer's disease. But most forms of it, it's not linked to a specific gene. There is some evidence, though there's not a lot, but it's growing, to epigenetics and Alzheimer's disease. So we know some environmental cues of Alzheimer's disease, meaning that often people with Alzheimer's disease have one or more of these environmental cues. These could be poor diet, lack of exercise, inflammatory conditions like diabetes, and obesity, as well as others, but these are the ones I'm just going to list right now. Now, some methylation studies have shown the following: that there is a general loss of methylation around the age of 70. And this is when we begin to see the plaque buildup in the brains. Specifically, we see reduced methylation of the APP gene and the tau gene, both genes that have been linked to Alzheimer's disease. Now, these next 3 points I want to make were made by looking at autopsies of people who had died of Alzheimer's disease. So there were methylation errors in the prefrontal cortex, which is linked to memory and higher cognitive thinking. Also in the, I'm going to say this wrong, entorhinal cortex. I'm sure someone will tell me if I said that wrong, but it's spelled ENTORHINAL, then cortex. This is also linked to memory. And as a control, they looked at the cerebellum, which has a role in maintaining balance and is not usually linked to Alzheimer's disease. And in the cerebellum, there were no methylation errors, as you might predict. But it was an important control to do, to make sure it wasn't just a generalized methylation problem. Okay, that's all I have to say about epigenetics and memory and Alzheimer's disease. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.